Hello, everybody. My name is Mark Cronin from Liverpool John Moores University in Liverpool in England, and I'm very pleased to be talking to you today about so-called in silico models for the prediction of toxicity. And just to explain my background, a little bit about my background, I'm just going to give you a synopsis of my career up till now. And we have actually just reached the 30th anniversary of me starting my PhD in this area. And my PhD I started on the 1st of October 1987 in what we then called environmental QSAR. And I took this PhD because since school and early in school, I was particularly interested in biology. And during my biology degree, I became interested in ecology and modeling large complex systems such as ecosystems. The opportunity came up to do my PhD in environmental QSAR, and I was very pleased to do this. And we were looking at prediction of toxicity in those days to fish and also to microorganisms and invertebrates. Since my PhD, we've been involved in very many projects, mostly externally funded, many through the European Union, through industry, looking at the prediction of harmful effects from chemical structure. And I've been lucky enough over the past 30 years to work with many different industries, in pharmaceutical industry, cosmetics industry, pesticides, and other industries, but also with government agencies in the UK and Europe and in North America and elsewhere, and also many non-governmental organisations, such as PETA, who are sponsoring this lecture. And naturally, since I started my PhD 30 years ago, I've seen many changes in science and in the world and in politics and in society. And these have given us many opportunities, which we've capitalized on in science and our projects, and much progress has been made. And thinking about this, two of the main differences in the last 30 years are, firstly, particularly that the connectivity within Europe that we didn't have in '87, that was before the Berlin Wall came down, and obviously the vast amount of new technologies. Particularly, I'm focusing here on the internet and internet facilities, and also the computational approaches to predict toxicity. So our learning objectives for today are to understand why we're going to predict and why we need models to predict toxicity and the philosophy behind that. I'm going to tell you a little bit about and show you some data resources where you can find existing toxicity data. And then I'm going to give you a very quick run through of structure-activity relationships and quantitative structure-activity relationships and how they can be used to predict toxicity. And lastly, I'm going to illustrate all of these issues with freely available, preferably internet resources. And I will probably say several times, but I will say here, I'm going to focus on freely available resources, but of course there are a number of commercially available resources as well, which may have more data or more sophisticated models or more support behind them. So the philosophy of in silico approaches, and clearly here, much of this is related to finding information out about chemicals without the need to test on animals. So let's imagine that you're faced with a scenario where you have to understand the risk associated with exposure to a particular chemical, but you don't have the facilities to test that chemical. So the first thing you would want to know is whether you knew anything about that chemical itself. So has it been tested previously? Can you gain access to those data? If there are no data or insufficient data for the chemical, can you find anything about similar types of chemicals and maybe interpolate across or make a prediction from that? And if that's not possible, can you make predictions of effects and toxicities directly from chemical structure? So I'm going to address each of those three aspects here, and there'll be some references along the way that you may be able to read up on, and also some websites that you can use these databases and tools freely. And lastly, it's very important that we use this information correctly. So I'm just going to mention that about the kind of issues we need to consider. So in Silico tools for toxicity, I've just summarized them here, that there are actually many others than what I've listed here. But firstly, we're going to talk about databases of toxicity data. And this leads us into an area called chemoinformatics. And chemoinformatics is the engine, if you want, behind the databases, the way in which the toxicity data are captured and the way in which they're presented, and you can search them and use them as a searchable tool. I'm going to mention very briefly a technique called read across, but that's going to be very brief. And then we're going to talk a little bit about structure-activity relationships and how we can apply them, where they're from, and what they can be used for. I'm going to give you a simple illustration of quantitative structure-activity relationships, QSARs, and then mention some expert systems. But as I said, there's many other in silico tools for toxicity prediction, and particularly we have areas and techniques for predicting kinetics, which I haven't got an opportunity to go in here, and also bioinformatics. So it is a very broad area with a lot of information. So the first thing you can do, if you don't know anything about a particular compound, is see whether there are any existing data. And the fundamental principle here is that if you've got existing experimental data, those are always going to be better than the type of predictions I'm going to talk about here. And there are many existing experimental data for chemicals. Not all chemicals have data, and these cover many endpoints. And one thing you'll have to do when you access these data is assess their quality. In order to help you do that, there are a number of freely available databases and tools exist to do that. I'm going to mention a couple of freely available databases and show you how they look and how you can extract information, but remembering that there are also commercial databases as well, which, as I alluded to before, may have many more data for different types of compounds. So the first thing to say is that if we're going to search for a chemical structure, then the chemical structure itself must be correct. And this sounds very, very trivial, but it is actually a real problem in many chemical databases, both commercial and freely available. And there's lots of issues here, for instance, dealing with stereoisomers and other types of isomers, how we consider impurities within a chemical structure. And also as well, fundamental issues such as whether we include the salt form. So for instance, if it's been formulated as a salt or it's been recorded as the chemical itself. To help us, of course, we have many kinds of chemical identifiers. And the obvious one you'd think of is using chemical names. But as I will illustrate in a moment, this can become complex. We have registry numbers through the American Chemical Society Chemical Abstract Service. These can be useful, but there are problems. Some chemicals have various different CAS numbers. Other CAS numbers are incorrect, and not all CAS numbers will be recorded. So typically, we may want to go to other kinds of identifiers of chemical structure. I'm going to focus here on the so-called SMILE strings and use those. I'm assuming some knowledge of those, although you may want to read up some more of them. And also, we have other more recent innovations, such as the INCHI keys. But I'm going to focus on SMILE strings. And at this point, software or programs, internet sites such as ChemSpider are a very useful resource to help us identify chemical structure. So ChemSpider is a website. Here is the front page of it if you go to chemspider.com. And a couple of things to notice about it. Initially, it's made freely available by the Royal Society of Chemistry in the UK. And they fund it as it's freely available, and they fund it through selling advertising. So be aware of that. You'll see here that at the moment, or when I produced these screen slides a couple of days ago, it had 59 million chemical structures in it, of which we had from 487 data sources. So an enormous amount of information. Of course, we don't have information on all, with the exception of the chemical structure, we don't have information on all 59 million chemical structures there. And indeed, we have data and properties for many, many fewer than those. So into ChemSpider, just to illustrate this, I'm going to illustrate the following slides with caffeine. So I've just typed in the name caffeine here and clicked on search. Click here. And this now leads me to a page where I can see the structure of caffeine, some basic chemical information, the molecular formula, molecular weight. And you have an opportunity to click through a number of options here. So I've just clicked on more details. And the first thing you'll see is the chemical names. And this is a fraction of the names that are available for caffeine. And this is one reason why it's very difficult to use chemical names to search, because what you'll see is there are chemical names. The name for caffeine is different in different languages. So what are we going to relate to? We have chemical names, we have CAS numbers, and we have all sorts of other identifiers here, including trade names and commercial names. So what is favoured to us then, rather than looking at names, is to consider, I mentioned the SMAL string and the INCHI keys here. And you'll see the SMAL string is written here. And this is just a 2D, one-line denotation of the chemical structure. And I can copy that into the browser or into my clipboard by just pressing here, the same as you would do if you press Control-C. We also have INCHI keys here if you want to use those, but it's true to say that most software uses the SMAL string. And if you look elsewhere, if you click on Properties in ChemSpider, you'll see all sorts of information that may be relevant to you. Here you've got experimental data, if you click and open these boxes, and here you'll even see some toxicity data. So it does act as a simple, trivial toxicity database. However, what you're going to want to do to find toxicity data is to go to more sophisticated and bespoke toxicity databases. So when you've confirmed your chemical structure and your identifier, such as a SMAL string, you can then go to one of these toxicity databases. I'm going to show you a couple of those, and these contain freely available toxicological data and other uses and other information such as properties and uses. I've got a slide later on with some of the bigger databases, but I'm going to start with one that's very dear to my heart, which is the Cosmos database. And again, we require you to register this with your email address, and it's free to use, and we just ask for your email address to keep you aware of alerts and updates. So Cosmos database was an output, and the URL is written here. It's very straightforward. Cosmos database is an output from the Cosmos project, which ran from 2011 to 2015. And was funded by European money from the European Commission and also the cosmetics industries themselves. And it was one of our main outputs. It contains about 44,500 unique structures. So put that in the context of the 57 million we had in ChemSpider, of which we have some data for about 1,600. So you can immediately see the kinds of numbers come down very quickly where we have significant data. In terms of toxicity, we've made a real effort to create an oral repeat dose toxicity data set here. And as we were funded and working with the cosmetics industry, we increased the number of cosmetics related chemicals. So if you go to the website, you'll find a page like this, and you'll see that I've already registered and I'd logged in here. And you will want to go to the option search the database. And that's what I'm going to do in a moment. You can also download data and run a tool for something called threshold of toxicological concern through the computational methods. So going back to database search, I've entered in here caffeine. We're going to have a look at caffeine again. And actually, as you enter it in, you should see the names start to appear or recognize that it knows something about caffeine. So you enter caffeine in there and press search, and it will start to find information it has on caffeine. And actually, caffeine has a lot of information in the Cosmos database. And you'll see a response like this. It actually says that it has 209 studies, toxicity studies on caffeine. It tells you a couple of the names. These are of regulatory importance, and some of the inventories. So ToxCast coding is the European Cosmetics inventory and so forth, and I'll allow you to investigate those. We have the structure, and to access data, we would click here where it says CMS 240, and CMS simply stands for Cosmos. So this is a unique Cosmos identifier for this particular substance. It has no other relevance whatsoever except an internal database issue. If we click on that, we then come to the page for caffeine in this case, and you'll see lots of information again, the chemical structure, but now more information on specific identifiers with regard to database and inventory belonging to. Moving on, if you enter the toxicity data and click on toxicity data, you'll see a tree open up in front of you. And as you click down here, I've opened up the oral repeat dose toxicity database, which is our own database of oral repeat dose data. We click going down through target organs to chronic tox data, and we have two studies here for mouse 90-day and rat 90-day. And I've just opened up, in this case, the information on the mouse 90-day study, and you'll see information on the dose level here and the output, so the lowest observed adverse effect level. And again, where we have blue, we can click on that, and we can find out more information. So this is now for this particular dose level for that 90-day rat study. We can see the relative effects and there were no changes. And again, remembering this is caffeine, we see that the lowest dose level, we see a decrease in body weight and an increase in water consumption. And anecdotally, that is probably quite obvious because caffeine can increase the metabolic rate of the animal and also increase the likelihood of dehydration. But we see no change at any other organs. And this becomes an interesting point. So is this now a toxicity? So is a decrease in body weight due to taking caffeine, can that be counted as a toxicity? And at this point, the data need to be analysed by an expert toxicologist in order to see whether this really is an adverse effect. Moving on to other types of databases, probably one of the most comprehensive is the OECD's, the Organisation for Economic Cooperation Development's eChem portal. And here is the website here. Again, this is now not quite so easy to use, but does contain an enormous amount of data. So if you click on the chemical substance search here, it will give you an opportunity. And again, I've entered the name caffeine to search for particular compounds, and you can use CAS numbers and other identifiers as well. And I've just clicked search. And now it searches a large number of databases from government agencies to try and extract data. And here you'll see below various types of data for it. So we can go and investigate this data. And you'll see there's fewer studies in this case, but from different sources. So again, we've gone from a quite user-friendly database with specific numbers of data into a data resource that searches many different databases and may give you more information about your compound. And eChem portal is very commonly used within the regulatory community to understand and predict toxicity effects. So imagine now we have a situation where you've looked for your chemical, but you've not been able to find it, or you haven't been able to find any data for it. So what you may want to do is search for similar chemicals and find toxicity data for those. So if there's no chemical or no data for your own chemical, what you may wish to do is enter your chemical. Here I've just entered into the Cosmos database. This is caffeine again, and I know I have data for caffeine, but let's see what similar structures we have. You have a function here, perform similarity search, and you can set whatever you want the similarity level to be. And we've got a similarity score out of 100. The higher it is, the more similar the compounds will be, is the basic issue. So I did this, and I find a large number of similar chemicals, the vast majority of which have no data associated with them. But there's two chemicals I was relieved to see had some data, and they're here. They're theobromine and theophylline. And actually, there are many data, 46 studies associated with theobromine. So if I'd have been in a situation where I had no data for caffeine, I might have been able to, and this is our molecule caffeine here, and this is theobromine and theophylline. I might have been able to use or look at these data to see whether they would be possible to what we term read across from one toxicity or one chemical to another. So looking at similar chemicals gives us a very good, or gives us a way of finding information out about other chemicals. As I said, this is called read across, and what the purpose here is to find chemicals that are similar to one another, and particularly chemicals which may have data and which we can, as the term says, read across. This is quite a famous diagram, and I do acknowledge Andrew Worth from the European Commission's Joint Research Centre, who first developed this diagram. It's been used by many of us many times. But what we're saying is, if we have a series of chemicals which we know are similar, and we have some data available for them, then we can begin to fill in the data gaps through this process of read across, sometimes also called interpolation extrapolation. And we're going to talk about the process of QSAR in a couple of slides' time. So to finish on toxicity data resources, there's lots of other places you can go. We routinely use something called the OECD QSAR Toolbox. The website is there. This is something you need to download and install, and is quite complex to use, but does contain a lot of toxicological information. There's two incredible resources now, ChemBol and PubChem, which both contain large amounts of information, and you'll see the numbers there in the tens of millions of compounds and data points. And these can contain all sorts of information, and particularly with regard to protein binding, receptor binding, and pharmacological activity, but enormous data resources. As a first stop for toxicity data, however, I would probably go to the OECD QSAR Toolbox, which is downloadable from there. And there are many other types of data resources that you'll use. So what now if you can't find any data for your substance and you're not particularly pleased or think it's reliable, the read across approach, then we can go and look at chemistry and see what that gives us. So there's been a long history of using and understanding chemistry and how that brings about toxicity. And we have some very widely and well-established techniques. And indeed, the QSAR principles that I'm going to talk about go well over 120 years history. These techniques have been used particularly in pharmaceutical pesticide industries, and more commonly, more recently, the cosmetics and personal product industries, for decades, in some cases 50 or 60 years. And we have many pieces of software to do this, some of which are freely available, and probably the more sophisticated ones do come with a cost and are commercial, or certainly do require payment of some sort. The simplest thing we can do is apply so-called structure-activity relationships. And these represent the relationship between some aspects of a molecular feature, and we'll see this on the next slide, and toxicity. And what we can do is use particular molecular features to separate out and classify compounds, as this little schematic says here. We can do it for lots of different endpoints. We can look at toxicity, and we can look at the absence of toxicity. We can look at absorption and other pharmacokinetic type parameters. And here's an example of a development of a structural alert. Here you have 4 compounds which are carcinogenic, and they're also highly mutagenic. And historically would have asked, why are all of these compounds mutagenic? And clearly the reason is that they contain the same functional group, in this case an aromatic amine group. And this is the basis of our structure-activity relationship. So the structure-activity relationship here is that an aromatic amine is associated with mutagenicity, which can lead to carcinogenicity. And we then want to rationalize what we term the structural alert for aromatic amines. So the structural alert is the presence of an aromatic amine on a molecule. And we can understand this in terms of what we know about the chemistry. So primary aromatic amines are known to undergo metabolism, and then metabolize the reactive nitrenium ion. It's the nitrenium ion actually that can bind to DNA, and we can understand and rationalize the organic chemical reaction there, which is a first order nucleophilic substitution SN1 mechanism. And we can actually see this happening. We can measure it by measuring DNA adducts by adding aromatic amines to DNA and using analytical chemistry to see the adducts in place. Here's the reaction mechanism. It's a 2-step reaction mechanism, N-hydroxylation and O-acetyl transferase. So in a moment, I'll show a slide that has a reference where the full chemistry of this and many other types of reactions is captured. So this is one structural alert for mutagenicity, in this case, the structural alert for aromatic amines. And historically, many other fragments and structural alerts are available for mutagenicity. And I really would like to highlight, this was a revolutionary paper in its time from John Ashby, who then worked at ICI, and Ray Tennant from the US NIH, that captured really decades of work in terms of electrophilic reaction chemistry. So in 1988, they published this, and this was their supramolecule, which contained about 20 electrophilic fragments, all associated with causing mutagenicity. Of course, this molecule does not exist, and when it was published, it was only available on paper. I remember receiving the reprint of this as a student. And of course, we've built this up into computational models now. So showing how this would work, here we've got 3 molecules, and if we look at our supramolecule that Ashby and Tennant proposed, then we should be able to see for each of them, the epoxide group is present there, so we would expect that to be mutagenic, the aromatic amine and the halide group there. So all of these molecules would be expected to show mutagenic properties, and indeed they are all carcinogenic. So what this demonstrates is that if we understand the structure-activity relationships, capture them through structural alerts, we can use this to predict toxicity. And of course, there are many types of fragments associated with DNA binding. And this is some work I published with a very talented organic chemist colleague of mine, Steve Enoch. And we defined many more of these types of chemistry. And if you go to that paper, you'll see that the chemistry defined. Here we see our reaction with an aromatic amine. So there's many types of chemistry here. And the point is that if you see these chemistries within a molecule, then you should be aware that they may be mutagenic, and this may prevent the need for animal testing. There's also a website called ochem.eu, And this is the home page, and it looks very complicated. And I don't suggest you try and read the whole thing. But if you go to this option here, screen compounds with tox alerts, what you'll be able to do is start using these structural alerts yourself. So we can input a molecule, search for structural alerts. So I've clicked on that, and you come to this screen, and it asks you what you want to do. So we just want to screen our molecules here. And then you get to a screen that looks like this. Now, at this point, you're going to have to enter in a SMILES string. So I've actually put in that compound we looked at before, the diaminobenzene. So this compound here, and you'll see the SMILES string here. If you want to use this, you're going to have to copy that SMILES string, and I've got it enlarged on another slide exactly. You then press load structure, and you'll see your structure appear here. Unless you see the structure appear here, then it's not going to work. And you can decide what kind of endpoints you want to look at. I've just chosen genotoxicity and mutagenicity and start screening. And you'll get an output. This is our output for the diaminobenzene, and it tells us what we know, of course, that it's an aromatic amine. And it's found, and it codes up those structural alerts from the Ashby paper. So it tells us what we already know, it's an aromatic amine. So if you click on that, then it gives you more information about the aromatic amines. It gives you some code about how it's coded, and you can go to the original reference and find out and read more about it. So it contains much more information than you would be able to normally look at. We can put compounds into it, and we'll search for any number of structure-activity relationships. So to summarize structure-activity relationships and structural alerts, the presence of an alert may indicate toxicity. However, we need to be careful. If you screen a compound and don't find structural alerts, it may indicate no toxicity, or simply that you need more information. Some types of toxicity are really useful, or can be really well parameterized in this way. And we've got really good alerts for genotoxicity and also skin sensitization. But we're still working in areas such as repeat dose and organ level toxicity. So this is widely used. These techniques, and particularly the commercial systems, are widely used for screening and prioritized molecules. And at the end of the day, if you get a good prediction that you understand, then there's no need to test that molecule. So far I've been talking about qualitative predictions, or yes, no, the presence or absence of toxicity. That's what structure activity relationships and structural alerts normally are. They're qualitative. But often we want to make a quantitative prediction of potency. So in other words, how relatively potent something is, such as an LD50 or an EC50. And here we need to go to a quantitative structure activity relationship, or QSAR. So I'm going to call these QSARs from now on. And these are designed deliberately to relate the potency of molecules to their molecular properties. So a QSAR can be defined as any attempt, or an attempt to relate statistically the biological activity of a substance to its physicochemical properties. So implicitly within that, these are models, and we need to base the models on a series of chemicals for which we have, in this case, toxicity data, and relate those data to the physicochemical properties and physicochemical structures. So I've got a very simple example just to run through here. And this is a real data set, they're real data. And the first thing to say is that we have a series of compounds. And I know a lot about these compounds. This goes back to my PhD work. And we range from aliphatic alcohols to ketones to esters, and even a bromo halide here, a bromoalkane here. We have some toxicity data, and these are data for acute toxicity, and they're actually acute toxicity to an aquatic species here. The data have been converted, so the lower the number, the less toxic it is. So the least toxic compound is methanol, methyl alcohol. The most toxic compound is tridecanol. And you'll see that the toxicity, the range of toxicity is over 5 orders of magnitude. And just looking at this series, you'll see that toxicity increases with chain length, or in this case, we parameterize that more correctly with hydrophobicity. So log P is the octanol water partition coefficient. This phenomenon has been known since the 1800s, that we increase toxicity with hydrophobicity. And of course, what we can do, we've got a nice data set, so I've just plotted the data out here. So increasing toxicity against log P, and predictably we get a straight line. So just to remind you, this axis is increasing toxicity from methanol up to the tridecanol, and this axis is increasing hydrophobicity as quantified by the logarithm of the octanol water partition coefficient. Of course, what we want to do is put a line through those data, and with a line we can put a line of best fits, and we can use techniques such as regression analysis to make that into an equation. So regression-based QSARs are usually very simple, and we can think of them in a generic way such as this. We can put any number of properties in. In the QSAR we've got here on the previous slide, we simply have one property, in other words, log P. And toxicity is this function, in this case, of the acute toxic effect, which is a millimolar effect causing a 50% concentration. And we've taken the inverse logarithm to get that number we saw. And typically, we would want to support the QSAR by measures of goodness of fit and productivity to understand how robust the QSAR is. So going to, again, just showing what this would look like. For our data set here, which I explained, we saw that this gives us this lovely relationship between toxicity and log P, and that our line of best fit is transformed into a relationship such as this. Our conversion of toxicity equals 0.75 log P minus 2. And again, remember there are units involved here, which I have defined as millimolar units. Log P we can calculate very, very easily using freely available software. And you'll see some figures here. So N says we've got 13 substances. R squared adjusted is the coefficient of variation adjusted for degrees of freedom, and S the standard error of the estimate. So they tell us that we have a good statistical fit. So we can use this, of course, for a further compound, hexanol. We do not have any toxicity data for this. I apologize, that should say data. And this is not in the data set itself. However, and this is very important, it is very similar to the compounds in the QSAR. So if you go back and look at the training set, you'll see it was made up of aliphatic alcohols from methanol up to tridecanol. So hexanol does fit into that series. We can calculate the log P at 1.82. Therefore, we can calculate the toxicity value here. And here are the units of toxicity, the negative logarithm of the millimolar concentration. So I would say to you that this is as accurate for this compound, for this particular toxicity endpoint. This would be as accurate as a toxicity test, and there is really no need whatsoever for this compound to test. This is a very simple QSAR I've shown you. They can get very complex. Here's one with 7 descriptors in it, and we needed more descriptors because we were looking at more compounds. You'll see 185 compounds, and B, the mechanisms were much more complex and moved away from simply being driven by hydrophobicity. So you will see very complex QSARs, and you will also see going into very multivariate and nonlinear statistical techniques such as neural networks, support vector machines, and we're now going into the areas of deep learning to try and extract information. So We prefer to use the simplest technique possible, but it really does depend on the compound, the type of toxicity and the endpoint. So you can use QSARs. Here's an example through the Vega Hub of some software called Vega that you can download, freely available, download. And I'm just going to show you quickly how you can use this. So you can download this onto a PC or a laptop, and it allows you to make a prediction using QSARs directly from chemical structure. Again, when you start it, you need to insert a SMILE string. This is the same SMILE string we had before. So the diamino benzene, and the uppercase and the lowercase are important here, just as a note. So you'd enter your SMILE string, you can press the plus sign there. You should see then the molecule appear here, the SMILEs and a depiction of the molecule. You would then go through to choose and select which type of information or which type of prediction you want. I just selected a mutagenicity prediction here. So we know this molecule is mutagenic, but we hope the predictions find that. You export the results by saving a PDF file and click on predict, and you will end up on your PC or laptop with a PDF that you will open and will give you a prediction. In this case, prediction is mutagenic, and you'll get a lot more information about the particular prediction here, which I'm not going to go to in any more detail. But if you use the Vega software, you will find that out. So another very useful website, again, the Vega software is from the Mario Negri Institute in Milan in Italy, and from the same group, is a website that gives you lists of the available software for all of our toxicological endpoints. So you'll see here skin irritation, eye irritation, mutagenicity. I just opened the website on mutagenicity, and the Vega software, which you may be able to see here, was the one I've just shown you. There's other kinds of software freely available, some of which are better than others, and it will also give you a list of the commercial software here. I haven't gone into any details on commercial software. And you will see the same for other endpoints, such as skin sensitization, repeat dose toxicity, also environmental endpoints. So this is a very useful resource if you want to go and find out what software is available to make predictions for specific toxicity type of endpoints. So to end with, I've talked through a number of different ways of firstly obtaining data, then using structure activity relationships through structural alerts, and finally QSARs. And I mentioned with the data that if you retrieve data, you have to determine as a user the quality of the data, and you may need expert advice on that. Just don't assume because it's in a database that it's correct. But in the same way for your structure activity or SAR or QSAR, as a user, you need to determine how good your model is and how appropriate it is for your compounds. So you remember with the QSAR, I said that the compound we made a prediction for, hexanol, was appropriate because it was similar to the compounds in the data set. And I have a lot of experience in being able to be confident in that, but that's something you can do through looking at the training set. With those things said, it must be said that careful use and appropriate use of in silico tools will replace animal tests and certainly will reduce the amount of animal testing that is required for toxicity. So to summarize my lecture, I've talked about some in silico approaches. There are many that can be used to help us understand and predict toxicology. We mentioned a couple of databases. I've given you some screenshots of those, and there are several more that you can use. We also looked at structure activity relationships, and you can go to freely available resources to use structural alerts to screen for potentially toxic compounds. And there's a large number of QSARs which can predict potency, and I've given you an illustration of one. And again, using these and other methods, particularly in combination and building up a weight of evidence with, for instance, in vitro and high throughput and high content data, we can eliminate the need for animal testing. With that, I'll thank you for your attention, and I wish you a successful course and an interesting course, and good luck in your careers wherever they may take you. Thank you very much.